Hi, I'm Krish, and in this week's online lecture we're going to be focusing on the basics of experimental design. We're going to be doing this in the context of a case study, and it should all kind of fall in line with some of the lectures you've been taking for the past 7 weeks. So when we're designing an experiment, there are many, components to how we get from answering our research question to then publishing and reporting our findings. So we start with a background, we then formulate a research question or a hypothesis. We then have an objective or an aim. We then think about a methodology, so how we're going to achieve our aim. And then we have to get our results, we have to analyse them, and then report our findings. And it all revolves around something called the scientific method. So the scientific method is kind of a process from how we get from our question to our end result. And we're going to be looking at this diagram quite a bit throughout this online lecture. So like I said, today's online lecture is focusing on a case study. We're thinking about a congenital abnormality called serenomelia. Now, the first thing we have to do when we have a research topic in mind is think about the background. So do a bit of background research. And when we do this background research, we begin by reading some online articles. And these can be things like review articles, which is a great place to start because they don't have a lot of methodology, so don't get bogged down in really complex methods. And then you can also read primary research articles, which is kind of the gold standard. And then if you're interested in perhaps some human data, systematic reviews are a great place to start because they collate a whole bunch of trials and data in the one place. So we need to think about what is known about the topic. So think about all the existing research, the gold standards of treatment perhaps, and what is known about if we have a gene of interest or a pathology. We need to think about what's not known about the problem, and then we need to identify that gap in knowledge, and it's that gap in knowledge that's going to be the focus of your research question. Now, you need to think about what specific aspect of the research problem you're going to address, because a lot of this research can be quite broad, so you need to narrow it down and focus on a certain topic. So like I said, today we're focusing on a congenital abnormality called serenomelia. And it essentially occurs when the lower limbs are fused together. And this occurs prior to birth. And it's typically called the mermaid phenotype or the mermaid congenital abnormality. And it's a very rare congenital abnormality and it occurs in about 2 in every 100,000 live births. Now, it appears when there's the fusion of both legs, and these newborns have a single umbilical artery instead of 2 like we usually do. They can also present with a few renal abnormalities or kidney abnormalities where they have an atrophic kidney, so that means it's a lot smaller than it usually should be. Cystic, so it has some small cysts on it, or completely absent. Now, I've done a bit of background research on this congenital abnormality by reading some primary research articles or some review articles, and what I found was that there's a few reasons why serenomelia might occur, and it could be due to an alteration in chordomesoderm development. It could be due to some mechanical defects that occur during birth, so when the amniotic sac, when the fetus is developing in the amniotic sac, some compression or folds of the amniotic sac could result in compression of the posterior region of the developing fetus. Or perhaps some sort of ischemic event, so when there's not enough blood reaching the posterior side of the embryo. So what's known about the genetics? So I've done some more research, so I've had a little look at some review articles looking at the genes that could be involved in serenomelia. And I found that there's perhaps 2 candidate genes. So the first could be twisted gastrulation gene. So mutation in this gene results in mice that are quite small and growth restricted. It also results in mice that have skeletal abnormalities or skeletal defects. The second candidate gene is BMP7 or bone morphogenic protein, and you might be familiar with this signaling protein as it's been mentioned in a few of your lectures so far. And mice that have a mutation in this gene, in both alleles, usually result in being perinatal lethal, which means that they don't survive throughout gestation and are usually born preterm or stillborn. Now, through looking at another research paper, I found that TSG and BMP7 were found to be expressed in the same regions of the developing embryo. So you can see up here, so in the first row, that BMP7 is being expressed, and then in the bottom row, TSG. And throughout the whole embryo, you can see they're being expressed in the same regions, in the same pattern. So this might suggest that they're interacting during development and could be involved in some sort of skeletal abnormality that contributes to serenomelia. So what's unknown? What don't we know about serenomelia? So what could be the underlying cause of this developmental defect? So we know that there could be two genes involved. So perhaps could the absence of both of these genes result in that phenotype of serenomelia? So it's now time for us to formulate a research question or a hypothesis. And that's the next step in our scientific method. We've asked our question, we've got a topic in mind, we need to do some background research, and now we need to formulate a hypothesis. So what is a hypothesis? So the textbook definition is a supposition or proposed explanation made on the basis of limited evidence as a starting point for further investigation. Now I've underlined 2 or 3 words there, and the proposed explanation is really important because we don't know if our hypothesis is true yet. And it's made on the basis of limited evidence because we need to have it based on some research. We can't just make a hypothesis on a whim. It needs to be based on some sort of foundation. So a hypothesis should be specific and it should be testable. It shouldn't be too broad, and it should be confined to a specific research question or topic, not the global problem itself. So it needs to be narrowed down and it needs to be feasible. We need to be able to test it. So an example of a hypothesis might be that genes involved in development may cause a congenital skeletal abnormality. Now this is a very broad hypothesis. Genes, we could be talking about any gene here, and congenital skeletal abnormality, that is a very broad term for a bunch of skeletal pathologies that occur during development. So it's not a very good hypothesis. A better hypothesis might be that knocking out both BMP7 and TSG will result in a serenomelia phenotype in the developing mouse. Now this has both of our genes of interest here. We're talking about ablating these genes or knocking them out. We're talking about perhaps a mouse model here, and we're also talking about the actual congenital abnormality we're interested in. Now, we also need to formulate an aim. Now, an aim is a bit different to a hypothesis, and it usually confuses a lot of people, but it's really simple. All it is it's the direction in which we want to take our study. It's our objective. So it's like our hypothesis in that it should be specific. It should be achievable as well. It should be within the scope of our reach. It should also be feasible. It's what we're setting out to achieve, and it's a bit different to our hypothesis in that it's more technically focused and focused on the methodology. You're explaining what you're setting out to do here. So an example of perhaps a not so great aim might be to determine if genetic mutations cause serenomelia. It's not very specific. We don't know which genetic mutation we're talking about. It's not a very good aim. A better aim might be we're here to determine if a double knockout, so getting rid of both BMP7 and TSG, will result in a serenomelia phenotype in the developing mouse. So that's the end of the first video. Head to Moodle, answer some questions, and I'll meet you back in the next video.